Hello everyone. Now let's take up our next topic from chapter 9, Strategies for Enhancement in Food Production. So today's topic is plant breeding for improved food quality. It is estimated that about 840 million people in the world do not have adequate food to meet their daily requirements. About 3 billion people suffer from protein, vitamins, and micronutrient deficiencies, or you can say that they suffer from hidden hunger. Why? Because these people cannot afford to buy adequate vegetables, fruits, legumes, fish, and meat. Their diets lack essential micronutrients like iron, vitamin A, iodine, and zinc. So, deficiencies of such essential micronutrients in the diets could lead to the increase of the risk for diseases. It could reduce the lifespan and also the mental abilities of a person. So, therefore, a new approach has to be followed, which targets at making crops with all these essential micronutrients. Such an approach is biofortification. It is the breeding of crops with higher levels of vitamins and minerals. Higher proteins are also incorporated along with healthier fats. This is the most practical means to improve public health. Earlier, fortification of the crops was also done. So, fortification and biofortification are different from each other because in fortification, nutrients are added during the processing of the crop, whereas in biofortification, the nutrients will be added in the crop itself, thereby increasing the vitamins, minerals, or essential micronutrients in the crops. Plant breeding is done for improved nutritional quality, and the various objectives of improvement are protein content and quality, oil content and quality, vitamin content, and micronutrient and mineral content of the plant. Proteins of cereals and millets are lacking lysine and tryptophan amino acids. In 2000, maize hybrids were developed that had twice the amount of the amino acids lysine and tryptophan. Atlas 66, which is a wheat variety, was also developed. This variety was having a high protein content, and it has also been used as a donor for improving cultivated wheat. An iron fortified rice variety was also developed. This variety contains over 5 times as much as iron as in commonly consumed varieties. The Indian Agricultural Research Institute, New Delhi, has also released several vegetable crops that are rich in vitamins and minerals. For example, vegetables which are enriched in vitamin A, like carrots, spinach, and pumpkins, were obtained. Vitamin C enriched bitter gourd, bathua, spinach, and tomato were also made. Bathua and spinach were enriched with iron and calcium. Protein enriched beans were also developed, like broad beans, lab beans, French beans, and garden peas. Along with this, number of other plants were also developed by IARI. So as we know that demand of food is increasing due to increase in human and animal population, even if the population shifts from grain to meat diets, it will not solve the problem because it takes about 3 to 10 kg of grain to produce 1 kg of meat by animal farming. More than 25% of human population is suffering from hunger and malnutrition. One of the alternate sources of proteins for animals and human nutrition is single cell protein. Single cell protein includes number of microorganisms which can be used as food. Some microorganisms like cyanobacteria and spirulina can be grown as food. It can be grown on an industrial scale as source of good protein. So approaches are made for its large scale productions. So microbes like spirulina can be grown easily on materials like wastewater from potato processing plants, straw, molasses, animal manure, and even sewage. This is done to produce large quantities of this microbe, which can serve as food rich in proteins, minerals, fats, carbohydrates, and vitamins. This biomass is used as food by humans. Incidentally, such utilization also reduces environmental pollutions. Certain bacteria, algae, or fungi are also used as single cell proteins. It has been calculated that a 250 kilograms of cow produces only 200 grams of protein per day. In the same period, if we take 250 grams of microorganisms like Methylophilus, Methylotrophus, this can produce about 25 tons of protein because it is having high rate of biomass production and high rate of growth as compared to a 250 kilogram cow. Mushrooms are also eaten by many people. They serve as a rich source of proteins and they are lacking certain types of fats. The large-scale mushroom culture is a growing industry. It makes it believable that microbes too would become acceptable as food. Single-cell protein is rich in high-quality protein and is poor in fats. Both high quality of proteins and low quantity of fats constitute good human food. The term single cell protein does not indicate its actual meaning because the biomass is not only obtained from unicellular microorganisms but also from multicellular microorganisms, like we just have learned about fungi also. Traditional breeding techniques, they failed to keep pace with the demand of the food, and also they failed to provide sufficiently fast and efficient systems for crop improvement. So there was another technology which was developed, is known as tissue culture. Now let's talk about our next topic, that is tissue culture. It was learned by scientists during 1950s that whole plants could be regenerated from explants. Any part of the plant taken out and can be grown in a test tube under sterile conditions, provided they are having special nutrient media. So general approach to this culture includes the separation of an explant from a plant. This explant is then grown in a nutrient media. It is kept in the nutrient media for some days. After few days, there will be the formation of an undifferentiated mass of cells which is called as callus. So this callus will grow further to form an embryo or the plantlets, and these plantlets will can be grown in the fields or the pots. So this is one of the most rapid methods of plant propagation. So different parts of the plants can be used as an explant. This is possible that from a single cell a whole plant can be regenerated. So this is because of one of the property of the cell, that is called as totipotency. The property of or capacity of the cell to generate a whole plant is called as totipotency. As it was told that explants have to be kept in nutrient medium, so nutrient medium has to contain certain carbon source like sucrose. It must be having inorganic salts, vitamins, amino acids, certain growth regulators like auxins and cytokinins. What are the advantages of tissue culture? One of the advantages that it is the rapid method of multiplication. Another are it involves propagation of because it involves propagation of large number of plants in very short duration. Therefore, this method of producing thousands of plants through tissue culture is called as micropropagation. Another one is that all the plants which will be produced by micropropagation will be genetically identical to the original or the parent plant from which they were grown. Therefore, all these plants will be known as soma clones. Many important food plants like tomato, banana, apple, etc. have been produced on commercial scale using this method. Another important application of the method is the recovery of healthy plants from diseased plants. Although the plant is infected with the virus, the meristem, whether it is apical and axillary, is free of virus. So hence one can remove the meristem and it can grow. We can grow it in vitro to obtain virus-free plants. So meristem is a localized group of cells which are actively dividing and undifferentiated, but ultimately it will give rise to permanent tissue. So therefore these meristem cultures can also be done. The explants commonly used in meristem culture are shoot tips and nodal segments. Scientists have succeeded in culturing meristems of banana, sugarcane and potato, etc. Next method is somatic hybridization. Scientists have even isolated single cells from plants. First, the cell wall of the plant cells are removed by digestion with a combination of actinases and cellulases. The plant cells without cell wall are called as the protoplast. Protoplast includes the whole of the components of the nucleus and the cytoplasm. So the technique involves isolation of protoplast from two different varieties of plants, each having a desirable character. So these two protoplasts can be fused to get hybrid protoplasts, which can be further grown to form a new plant. Fusion of protoplasts can be done with the help of chemofusion or electrofusion. The fusion of protoplasts not only involves the fusion of their cytoplasm but also their nuclei. The fused protoplasts are then allowed to grow on culture hybrid cells, and this will give rise to callus. Soon they will develop their own walls, and they will be now called as the somatic hybrid cells, and the process will be now called as somatic hybridization. Many plants are obtained by somatic hybridization. One of such plant is pomato. Pomato is a somatic hybrid between potato and tomato that belong to two different genera. The basic technique remains the same. There is the fusion of protoplasm, hybrids will grow, and the hybrid callus will lead to the formation of hybrid plants. So this is the picture of tomato along with the pomato. So the hybrid plant bears both fruits and tubers of the two parents. Unfortunately, this plant did not have all the desired combinations of characteristics for its commercial utilization. Tissue culture and somatic hybridization offers vast potential for manipulation of plants in vitro to produce new varieties, and all these measures enhance the production of food. Students, I hope you have understood all the topics of this chapter. Now let's go for the assignment. Try to answer these questions. First question is, a sugarcane plant has been affected by virus. How can a virus-free cane be developed from it? Why are plants obtained through micropropagation termed as soma clones? Name 3 food plants that are produced on commercial scale using this method. Write 2 advantages of micropropagation. IARI has released several varieties of crop plants that are biofortified. Give 3 examples of such crops and their biofortification. Why are proteins synthesized from spirulina called single cell protein? I hope you all can answer these questions. Thank you everyone.